Dr. Pepling, Dr. Melissa Pepling, is professor of biology at Syracuse University. Her lab specializes in the study of oocytes, hormone signaling, and ovarian organ culture. And while the other studies discussed today used the gas sevoflurane, which, by the way, is the most, I believe, the most popular anesthetic inhalational agent used today, Dr. Pepling used halothane, which was the most popular inhalational general anesthetic gas used in the 1960s and the 70s, and I think into the 80s, if not later. So halothane could potentially have much more of a legacy effect than sevoflurane, just for all practical purposes. But anyway, turning it over to you, you can share your screen, Dr. Pepling. There you go.
Okay. I want to thank the organizers for giving me this opportunity to tell you about some of our work and going in a new direction. And we're looking at in utero halothane exposure effects. The talks that went before me on sevoflurane were awesome, and I've gotten a lot of great ideas. Our work on halothane is at a much earlier stage, but there's some interesting parallels that you'll see as I talk. So as I said, this is a new direction for our lab. Our lab usually focuses on female infertility, and this is a major problem because about 12% of women of reproductive age suffer from impaired fertility. And one of the disorders is primary ovarian insufficiency, which affects about 1 in 100 women and is associated with an abnormal depletion of oocytes or depletion of the ovarian reserve, which we're very interested in. And there's a cartoon at the bottom of the slide that shows a functional ovary with lots of oocytes, the purple circles, and one of them being ovulated here, and then a very dramatic contrast of the oocyte depleted ovary with no oocytes, and of course that individual would be infertile. And so just one more slide about oocyte development. And what we're normally interested in is how the primordial follicles form. So oocytes, which are the green cells, develop in these clusters called cysts, which separate, and then individual oocytes are surrounded by granulosa cells, and that forms primordial follicles. And we're very interested in the regulatory signals that work to generate the primordial follicles. But for today, now I'm going to switch gears and tell you about our new interest in reproductive and developmental toxicity. So, reproductive toxicity can adversely affect reproductive functioning of both males and females, and also cause developmental toxicity in their offspring. And one of the most famous examples is diethylstilbestrol, or DES. And this was used to treat miscarriage for many years. But instead, it didn't actually help miscarriages, but instead caused effects on the offspring, especially in the females, adenocarcinoma of the reproductive tract. And that was discussed in one of the previous sessions, in session 5, if you want to go back and look, but that's interesting. But now, as this session, we've been focusing on anesthesia, which of course is necessary, as we've heard in some of the other talks. And in fact, about 2% of pregnant women in the US will undergo anesthesia for issues unrelated to gestation. And as you heard all the talks about sevoflurane, I'm going to focus on halothane, which was the general anesthetic, as Jill said, that was popular in between about 1950 up to the 1980s. And then I also want to bring in autism. So how is anesthesia exposure related to autism? Well, we've heard a little bit about that, but I'll just review. So there's a lot of case histories where the parent was when an autistic child was in utero when the mother underwent surgery. And this was a lot of these case histories are the pregnant mother, it was back in the 70s when halothane was used. And that's why we want to focus on halothane. And our hypothesis is that the general anesthesia exposure in utero affects the programming of fetal germ cells, leading to neurodevelopmental defects in offspring derived from those germ cells. And the talk you just heard discussed some of that, and we'll talk about what halothane does here. And this is just to review that here's the gestating mother. We're calling that the F0 generation, and then the developing fetus, we're calling that the F1 generation. Sorry, the gestating female was F0 generation. The fetus was F1 generation, and then the germ cells are also getting exposed, and that will give rise to the F2 generation. And so how could the anesthesia affect development? And one of the ways, and you've heard about this already, is by modifying the developing epigenome. So, and as you know already, I'm preaching to the choir, I know. The epigenome is a collection of chemical modifications to DNA or else to the associated proteins that affects gene expression. And so here is a cartoon showing the DNA in blue and showing that DNA methylation can occur to the DNA. And then also the DNA is wrapped around the histones, which form nucleosomes, and the histones can also be modified, which our studies have focused on DNA methylation, but it would be interesting to go back and look at the histone modifications as well. And then, so here, usually with DNA methylation, it's the cytosines that get methylated, as I've shown here. And so in genes that are unmethylated promoters, shown here for gene A, those genes are often active, whereas when there's when a lot of the cytosines in the promoter of a gene is methylated, then the gene is inactivated. So, and then we're going to focus on a little bit more on what's happening to the germ cell methylation, which you just heard in the previous talk, and I'll just quickly review. So this is looking at methylation again, and here's the developing embryo. The primordial germ cells form quite early, which will go to either develop into oocytes or sperm. And so if you look at just the primordial germ cell methylation during development, the methylation is erased. So here around E13.5 in the mouse, or week 6 to 19 in humans, the methylation is very low. And just like the previous talk, we also did our exposure at this early time point, which I'll come back to the specific time in a minute. Anyway, and this is just to remind you that the exposure to a toxin in utero affects 3 generations directly, that F0, the F developing fetus, the F1, and then the F2. And the F3 generation is the first generation that's not directly exposed. And that's the transgenerational effects, which I'm not going to focus on in my talk, but I'd love to look at that in the future. And then, and of course, epigenetic dysregulation could elucidate the biological origin of certain illnesses and disorders, including autism and autism-like disorders. So the project goal was to determine if in utero exposure to the halothane anesthesia affects the methylation profile of germ cells, and if this aberrant methylation could cause intergenerational behavioral effects. So our first aim was to look at the exposure, excuse me, if the exposure alters behavior in the F1 and the F2 generation. And I'm only going to present our data looking at the F2 generation. And then also, as I said, we're going to be assessing, we assess the regions of the genome that are differentially methylated on exposure to halothane, focusing on the gene promoters. So this is the study design. So we looked at 14.5 DPC, which is when the methylation, as I showed you, is very low, and we used exposed to halothane. And so that was the F0 generation. And then we took halothane exposed males and halothane exposed females, the F1 generation, and we did behavioral testing. We also did bisulfite sequencing to look at methylation status. And then also we did breeding to generate the F2 down here. So males and female, we crossed the F1 halothane exposed animals to unexposed animals, and then to generate our F2, which were either the, we looked at male and female for both if their father had been exposed in utero or if their mother had been exposed in utero. And then for all of these groups, we did behavior testing. So back to our AIM-1, looking at the behavior first, we looked at, as I said, we looked at both the F1 and the F2. The F1 actually didn't have any effects in the tests that we did, unlike the other studies that were shown, but there were some effects in the F2 generation, which I'll show you now. And I'm also going to give you a little bit more information about our behavior testing. So we use the elevated plus maze, which was used in some of the other studies presented today, which evaluates anxiety level. And we chose both this test and the other test that I'll tell you about because these are some of the autistic-like behaviors. For example, anxiety is often higher in autism and autism spectrum disorder. And so we looked at this and this is just, I want to show you the actual equipment that we use. This is the elevated plus maze, which has an open arm and then a closed arm that has walls on the side, and the mouse is placed on the apparatus, and then for 5 minutes it can freely roam around the apparatus, and we have a camera set up overhead that's recording the behavior and recording the time the animals are in the open arms versus closed arms. And the more time the animals spend in the open arms, the less anxious they are, and if they're more anxious, they're going to spend more time in the closed arms. This is just showing from another view. And then this is just the camera, how it's set so the camera can record the time in each of the arms. So this is looking at the F2 animals whose fathers were exposed in utero. And so on the left side, you can see we're looking at the males, and on the right side, the females. So for the males whose fathers were exposed, spent significantly more time in the closed arms, so they're demonstrating they have more anxiety. And then, but we didn't see that effect in the females. Then looking at the offspring whose mothers were exposed in utero, we see no effect in the elevated plus maze and how long they spend in the closed arms and how long they spend in the open arms, either the males or the females. So our other behavior test was using the social approach test to test sociability in mice, because autistic individuals often have reduced social ability, issues with communication. And so in this case, we used the 3-chambered social approach, which was also used in the previous study. And so here on this, we have 2 sets of 3. So this is the 3 chambers. The 1 chamber is where the mouse is placed into the chamber, and then there's a side where there's a mouse in this cup here, and then on the other side here, there's an object underneath that cup, and the mouse is placed in a center chamber with the doors closed for 10 minutes to acclimate, and then also is allowed to, then the doors are open and also for 10 minutes is allowed to acclimate to the 3 chambers. And then the mouse is placed back in the middle, the doors are closed, then the doors are open, and for 10 minutes, the mouse is, we record where the mouse goes using our software that's above the camera that's recording, and see how much time that mouse spends with the mouse in the mouse chamber versus how much time the mouse spends in the object chamber. And then I'm just going to show you a movie, if I can. I have to stop my laser pointer to do that. And this is just showing the mice, the actual experiment with the mice running around, and this mouse on the left is interacting with the mouse in that cup. It's very interested. The other mouse seems to be staying with the object, but this is just a subset of that time. And then I'll show you the data for that. So here we're looking at offspring of exposed fathers again, and the males on the left and the females on the right. And the males, there's no difference in the amount of time, either control or halothane, or males from the halothane exposed fathers. There's no difference in the length in either chamber. However, we did see something interesting and unexpected with the females, and that is the females from the treated fathers seem to have an increased sociability compared to controls, an increased time in the mouse chamber, which is very surprising. Anyway, and I'll summarize that a little bit more at the end. And now I'm going to show you the offspring of the exposed mothers, and we didn't see any differences between control and halothane for either the males or the females whose mothers were exposed. So now I'm going to go on to the second aim of this project, which is to look at which regions of the genome are differentially methylated when exposed to halothane, and focusing on the gene promoters. And we used reduced representation bisulfite sequencing, which was used in one of the previous talks looking at sevoflurane as well. But I'll just tell you about the method in case you're not familiar with it. So it's a method to assess DNA methylation, and it's like another technique called whole genome bisulfite sequencing, but it's more cost efficient, and also it's more focused on the gene promoters. And that's because it uses the restriction enzyme and also size selection to isolate these CPG islands, which are basically the promoter regions of genes. And also on the right here, you can see what happens is that any C that's not methylated gets converted into a uracil, but any cytosine that is methylated stays as a cytosine, and then this gets sequenced, and you can compare we compared the control to the halothane treated germ cells. And let me tell you more about that. So we collected genomic DNA from both halothane exposed and control mice, and we had 2 biological replicates for conditions. So we collected sperm from both control and F1 control and treated animals, or exposed animals, I should say. And then we also we wanted to collect oocytes, but we couldn't get enough material to use that for the sequencing. So we instead collected the whole ovary from both control and halothane exposed. And then the samples were sent to the Cornell Epigenomics Core for the sequencing, and then the data returned to us. And the other thing I should tell you here is that it ended up that we didn't have enough statistical power when we separated out the sperm and the ovary, so we combined together. So we just have basically 2 groups, control and then exposed, either germ cells or ovary, with the data combined together. And we hope someday to go back and to look at the oocytes separately as well. So what did we see? So this is showing differentially methylated CPG sites, and there were over 2,500 differentially methylated sites in this study. And by far the majority of them are increased in methylation. We're not sure why that is, but there's way more increased methylation, and only about 435 that were decreased in methylation. And then when we looked at just the gene promoters, combined those CPG sites, what we saw is that about 41 promoters had increased methylation and only 3 had decreased methylation. And I don't have the details, but I'll just give you a little bit of a summary of what we saw. And of course, the most interesting is that there were 4 genes that we found that were differentially methylated that are involved in nervous system development and behavior. But we also found some other interesting things: 5 genes that were involved in olfactory function, and 7 genes that were important for immune system function, which is very, very interesting because one of the talks, I think it was Dr. Martinek, found the same thing with his RNA-seq data with the sevoflurane. So that's kind of interesting. And then my favorite topic, reproduction, there were 5 genes that were linked to reproduction. Anyway, so I'm going to summarize now. So the F2 males from fathers exposed to halothane in utero showed increased anxiety in the elevated plus maze. And then the F2 females from the fathers exposed to halothane in utero demonstrated greater sociability in the 3-chambered social approach task. And this is very interesting that both of the effects we saw were only from offspring of exposed males, which is interesting because of the studies with the sevoflurane, where a lot of times the males there's more an effect on the male and not the female. Very, very interesting. And then also methylation, as I just showed you, was altered in 44 promoters from the in utero halothane exposed germ cells. And so some of the things we want to do now are to follow up on the genes that are involved in nervous system development and behavior, to learn more about these effects on halothane and how this could be transmitted. And then also looking at halothane exposure at different windows of development. And also our exposure was only for 30 minutes. And I would really like to do, after hearing the talks today, really like to do a longer exposure at the same time. We also would like to do some more behavioral tests. We had planned to do more tests, but COVID hit. And then we would be also interesting to perform the RNA-seq alongside the bisulfite sequencing to really directly examine effects on gene expression. And so I'm gonna stop there and give some acknowledgements. This work was all done by a master's student, Maggie McCoy, whose picture's up here. And then she was assisted by Lindsey Banks, another master's student who helped with all the behavior tests. And then she was also helped by the lab technician, Sue Getman, who helped with the halothane exposure and also all the breeding of all these mice. And then another lab at Syracuse University, the McDonald lab, 2 graduate students, Nico Wagner and May Ribeiro, also gave us lots of advice on how to do the behavior testing, because we have had no experience in testing behavior. And then also to analyze the bisulfite sequencing data, we had help from several individuals at Syracuse, including Yasser Ahmed Brahma, who's a new assistant professor, and then 2 PhD students from other labs, Caitlin McDonough and also Leanne Kelly, helped us a lot with analyzing that data. And so I'm going to stop there and take questions. And thanks for your attention.
Thank you so much, Dr. Pepling. Yeah, this goes to the power of having real meetings with real live people in a real place. I met Dr. Pepling at a Cold Spring Harbor germ cells meeting a couple of years ago. Back in the day when we had real meetings.
Oh yeah, that was the most important thing that I didn't say, was that all this work was funded by the Escher Fund for Autism, and by accident I met Jill at that meeting, and here we are. So thank you so much, Jill.
Somebody studies oocytes. Oh my God, I was so excited. Okay, so let's go to these questions, and then I have one, of course. Are there any SNPs related to such phenotype, or only affecting the epigenetics of the embryos? Also, have you looked on the extracellular DNA during pregnancy before and after exposure of the pregnant mice?
So the extracellular DNA during pregnancy, we have not looked at that at all. Interesting. I don't know about that.
I don't know. I'm just reading the question.
And then also we haven't, I don't know if there's SNPs related to the phenotypes at all. We've only looked at the methylation.
All right. Also, as a follow-up, does the mitochondria in the sperm affect anything at the F2?
I also don't know that. We didn't look at that.
Yeah.
So.
I don't know if some of our other speakers are still online. You can unmute and maybe say if you've seen effects of general anesthetic gases on mitochondria generally, and or mitochondria in the germ cells. Feel free to unmute if you have an answer. Nobody seems to have an answer. Actually, that's a question I got recently when I gave a presentation. All right, I have a question, because you study fertility and infertility at least in females. And again, this is just something I seem to notice. It may or may not be true. But when a parent has a significant history of surgery, I seem to see what seems to be increased rates of number one, infertility, and number
Mhm,
two, increased rates of miscarriage,
mhm.
even if they're technically fertile. Um, and then I saw one study. I was trying to figure out like what in the world could these gases have to do with infertility. Number one, I saw a lot of literature on DNA damage. Number two,
Mm.
I found one study showing that halogenated agents in general damage Sertoli cells.
Oh,
So I wonder if that could also be true with respect to the halogenated anesthetic gases and also with respect to the granulosa cells
mhm,
in the females. Because if you're damaging the support cells, you're going to increase,
mhm.
obviously, the rates of infertility. I'm wondering if you noticed anything with halothane and fertility.
Like, no, I don't think we did. I don't remember there being any effects, but I know that sometimes with I think there's studies with halothane that say that you have and um, that might have been in the first talk. There's preterm birth. So I'm pretty sure that halothane could probably affect that. Well, and as far as miscarriage, I wonder. I haven't seen anything, but I wonder if the halothane somehow is affecting the uterine. The uterus is supporting the developing embryo, so somehow it's affecting the placenta, or I don't
Yeah.
know. It could be
Well,
very.
yeah, I mean, it's actually recorded that halothane exposed to nurses in the operating room, nurses
Oh,
would,
uh,
at increased rates, experience a spontaneous miscarriage.
and then and
So,
then and we haven't. I mean, we haven't looked at the. I now I want to go back and expose animals and then look at their germ cells, like because I'm sure that if it affects their Tolley cells, it affects the granulosa cells. We haven't noticed that, but it could. Um,
well, this was just on halogenated agents generally. It wasn't
uh,
on halothane or any of the particular
okay.
anesthetic gases, but I think it's an important
Uh,
question because if you're giving a little kid one of these gases and you're damaging, the Sertoli cells or whatever, then that could have long-term reproductive effects, among
right.
other things. And then there's the DNA damage issue as well. Okay.